I found this course is something of a journey. And one of the sets of discoveries that I'm hoping to make is more where we learn not only about the science, because the science, has come out of your textbooks, but something about the people who are involved with it. And this is one example of that. We've had lots of others in the past few lectures. So I hope that that's a goal that you're willing to share with, enjoy with me. What I want to do is start talking about the sort of what we think of as the team of Schleiden and Schwann, who are generally given credit for the concept of the cell theory. And what I thought when I started working on the course, I really thought this would be a chance to find out a little bit more about who these people were and what their interests really were like. And as you go through them, what you'll see I'm doing is I'm trying as much as possible to use some of their texts and some of the material that they wrote so that we can get some idea of the way they think and what they're doing. I want to start with Schleiden because we always think of them in terms of Schleiden and Schwann. You'll see that they're actually pretty close together in terms of time and what they had in how they knew each other. They were actually good friends. But we'll start with Schleiden. And there are some interesting things that I've been able to dig up. Schleiden started out with legal training and then got started as a lawyer and then apparently had a difficult time, so difficult that he actually attempted suicide. And then eventually got involved in studying natural science, going from one German university to another. He traveled around quite a bit, but he did spend a certain amount of time at the Museum of Anatomy in Berlin, which is where he met Theodor Schwann. But he also ended up, and it's not in here so much, at the town of Jena, Jena, which has a famous university. Let me add this to the notes. In Jena as well. The reason that Jena matters is, as you'll see later on, probably, well, we may hear a little of it today. Jena is the place where Carl Zeiss set up his microscope company. And so there was actually a connection between Schleiden and Carl Zeiss. So we will probably get to that a little later. At any rate, as a botanist, he became very interested not only in sort of classical botany, which meant classifying and describing lots of plants, but thinking even more broadly about how plants fit into the structure of all the living material we knew about at that point. And also he became very interested in what you would now call the embryology of plants. As a result of all this, he ended up generating some very important documents, and I want to show you at least a little bit of some of them. I have to point out, this is just a characteristic that we benighted folks have to deal with, which is that although Schleiden wrote all of his papers in German, he was German originally, and his papers were written in German, each one of them has been significant enough that it acquired, if you will, a translator who converted this into English and actually in several others. So his works were actually pretty widely distributed in a certain way. At any rate, one of the things that came out of his study on what he called phytogenesis, that means how plants formed, how they develop, came out almost at the very beginning of this paper, which is called Contributions to Phytogenesis, in which he sort of reaches in as sort of a philosophical perspective and says, more or less, we have to think a little bit about whether we're talking about individuals or community. And it applies in animal wildlife, it applies in society, and it applies in plants. And what he found out, what he worked on, was that the idea that, in fact, okay, there are some cells that we sort of know about, some small molecule plants that we know of that are actually single cells. What he calls fungi, algae, the very lowest orders of plants are already, but then he comes in, he gets this other very interesting concept that every plant developed at any level of sophistication, if you will, or complexity, is an aggregate of fully individual, I'm not going to be able to draw this very well, fully individualized, independent, separate beings, each one being a cell. And then he goes on to talk about this, to say basically, look, every cell leads, quote, a double life. It has an independent one pertaining to its own development and its own structure, and another in which it becomes part of a plant. So what he wants to make clear is that it's very easy, given thinking like this, that the individual cells are the indispensable fundamental structure of the plants. Okay? Then he raises the question of how does this particular little organism, since he now sees it as being a separate structure, how does this particular organism originate? So let's see what he does, what he digs into. First thing is, I had to show this because I want you to get a sense that there is a certain sense of continuity. So within this same introduction, he says, it was Robert Brown. Remember Brown? It was Robert Brown who, with his comprehensive natural genius, first realized the importance, and in this case, he's talking specifically about, where is it, an opaque spot, which he called, that is Brown called, the nucleus of the cell. Okay? And so he gives Brown the credit for seeing the nucleus initially, but then he goes on to show that he's actually finding the nuclei sort of everywhere. So we'll get a kind of a, he's giving it a new name, which you'll see is maybe something of an issue, which he calls it a cytoblast. And you'll see why in a few minutes. But he goes through and describes in quite a bit of detail in this paper what this nucleus or cytoblast looks like. And one of the things that I was struck by in reading this, he points out that it's colored, that is to say, it's stained by iodine. Now, I remember when I was just getting started in school, one of the things we were given to look at with sort of cheap toy microscopes was actually the thin layer of material that you peel off an onion, not the dried out surface, but if you separate the individual segments of an onion, you can peel off a very thin layer. And when you do, that thin layer of material, you can actually see it divided up like this. And I remember, it's funny that you should remember these things, but I remember that I was told that if you stain these things with iodine, you'd see this structure of the nucleus in each one. Now, turns out We can't do that anymore in this sort of casual way because people don't use iodine as an antiseptic anymore. As I was growing up, that was the standard. And then it was replaced by mercurochrome, which is now something that people worry about, and additional kinds of antiseptics. But iodine was one of the common things. And I remember this idea that nuclei in an onion skin cell could be viewed that way. Very interesting idea. So what does he then show? So he then starts looking at what goes on in these plant cells. And he almost implies, but it's a funny kind of implication. He doesn't make an absolute statement that all the cells in the plant are individual units, but you saw that that statement that he started with here more or less implies that. Not here, here. No, back here, okay? That their individual cells are a critical component of what constitutes plants. So then he goes in to thinking about how this thing works. And basically he more or less says, look, this is what plants must be like. The plant grows because it has a number of cells that it increases. That is, it produces, an interesting phrase, the number of cells allotted to it, but it actually increases that number all the time, right? Because the plant, as he says, unfolds itself by the expansion and development of the cells that are already formed. And this, he says, is characteristic of cells as opposed, say, to crystals. People knew that crystals grew, right? You could get a salt crystal that gets larger and larger, but it's a very different process. In this case, he's talking specifically about things that relate to plants. And he says even more, not necessarily in animals. We'll get to that in a bit later, like next lecture, okay? But at any rate, what he says is you get this expansion by somehow increasing the number of the cells. And then the walls of these fully developed cells become thickened by the deposit of new material, and eventually they become hardened and more rigid. And that's where you get what he calls the lignification of the plant. So in a sense, this is the core of his contribution to our understanding of biology. That is that plants consist of units, each of which has a nucleus. And that nucleus then serves as a sort of, at least a marker for the individual cell that makes up that plant. And so the idea is that plants are made up out of individual cells. Now, here are just some illustrations that come from that original paper. And so what you can see here are drawings. I've put this thing on its side so that it would fit into the aspect ratio of these images. But you can see every one of the cells that he's drawing, even when they start small and get larger and larger and larger, okay, always have within them a nucleus. Like this, okay? And all the others that you can see here, even something like this, where you can make out there's a nucleus in the wall of that cell. And some of the stranger ones also have nuclei. So the nucleus becomes a constant and a marker for the cell itself. He became, among other things, a very popular lecturer. And he lectured all over the place, apparently, and collected these lectures into a book. And this book is indeed called, in German, Die Pflanze, but in English, The Plant, a Biography. This is a 600-page document by the time all the copies are done. So he gives a rather wonderful introduction. He gives a section called The Eye and the Microscope, in which he talks quite a bit about the whole idea of perception and how we get to see what we see with our eyes, as well as what we eventually see with the microscope, and the way that what we're doing is changing the image that our eyes receive when we use a microscope. So there's a chapter called The Eye and the Microscope. And there are then lots of details that go on about plants in general. And in one of these, he talks about the history of the vegetable world, in which he starts to speculate about where do things come from. Again, this is a man whose curiosity seems to be extraordinary as I go through his writings. So here's what he had to say. First, this is his comment from the eye and the microscope section. You have to really read, you get a certain sense of what his point of view was like, what his personality was like. Because here, he starts saying in this chapter, I've just copied over certain excerpts of it, okay? And what he says here, for instance, is in the 18th century is what he's referring to at the beginning of this passage, okay? The microscopes that were developed weren't that great, as we've talked about. They had lots of problems with them. He said, nevertheless, people became interested in what he's calling the elegant forms that they were able to see through the microscope, and didn't really care about the use of the microscope towards science. So he suggests, with a certain kind of snark, I mean, this is what I'm really amused by. He says, look, this guy Ledermiller wrote a book called Microscopic Delights for the Eye and Soul. And then this other one called Insect Recreations. And he says that even so, there were still people who were serious about this stuff, but even there were issues. And he raises this very interesting story about Swammerdam, which we really didn't discuss much. But towards the end of Swammerdam's life, I think we did discuss that he got caught up in what turned out to be a religious cult, a woman that was deeply invested in her religion and brought him into it and caught him so that, for instance, he was unable to publish some of his work until she had included a whole bunch of comments about praises to the Almighty and appreciation of heaven through these images that he got. And in fact, he destroyed, Swammerdam, a great deal of his notes because he felt that because you had to do complex things to see them, you were profaning God's mysteries. And so Schleiden is more than a little unhappy with this. He points out that if this were your idea, that you can't probe the nature of the structure of nature itself, you'd oppose every advance of humanity. So then he raises his own flag and he says it's reserved for our country, our century, to apply the microscope to its true use. Okay, and then he goes on to talk about the improvements in the microscope and, of course, his own observations. Now, one more little bit of snark, and then we'll get on with a little bit more of what he came up with. Okay, so he starts by saying, and I've had to cut off part of the document. He says, well, I don't want to really detract from Linnaeus because he really did a remarkable job. We haven't discussed Linnaeus at all because, as you'll see, Linnaeus wasn't much of a microscopist. But what Linnaeus did was he saw the organization of biological material in terms of what we now consider the Linnaean categories of phylum and species, that sort of structure that Linnaeus was able to lay out. And apparently, it says here that he is the author, nevertheless, of one of the, this should be an S, of one of the saddest of prejudices, which has long kept botany in the lowest condition, because he objected to the use of the microscope for the same sort of reasons, okay? And once again, he said it made it very difficult for the work that some people, and he quotes Malpighi, who did a lot of the work as well in the insect anatomy, that they had a long struggle to get very close to this kind of work, okay? Well, it turns out in the book that I will not be able to spend too much time on, there's a book called Botany as an Inductive Science, which I listed at the front here, which is again an enormous botany text that goes from the very simplest ideas of the chemistry that plants live in all the way through each of the individual families of plants. It's an enormous comprehensive book. But in the English edition, in the index or the appendix, there's a wonderful discussion about the use of the microscope because he considers it an essential tool. And I'm going to mount that, to put that discussion on Canvas so that you can read it. I hope you'll take the time. You can skim through because some of it is kind of too much in the sense of philosophical, but a great deal of it talks about the practical aspects of using a microscope, what you have to do to prepare a sample, what you have to do to think about what it is you're seeing. He spends a lot of time on the idea that if you just, there's a wonderful example, I'll mention this. He says, if you simply gave your sample to an artist to draw, and you may remember when Levenonc did that and others did that, the artist that doesn't know what he is looking at will simply portray what's there if the artist is very good, but won't be able to introduce a sense of what's the three-dimensional structure, what is the deeper structure, how does this entire unit fit together into a three-dimensional object. You remember that one of the things that made Robert Hooke's drawings so impressive was that because of the way he looked at the samples, he introduced into his drawings not simply a flat perspective, but actually a true kind of three-dimensional concept of what his objects were. So it's part of the article that Schleiden writes about what you have to do to really be a proper microscopist. And just because it's fun, this picture over here is the beginning of section 2. But I couldn't resist this sort of image of the tools of the microscopist at that point, because it introduces two types of scopes. It introduces the compound scope, which we're pretty comfortable with, but also very clearly includes the sort of single lens scope that Robert Brown used. Here's one more thing that came out of this analysis. This is in lecture 11 of that book. So we're talking about here, the history of the vegetable world. And again, Schleiden has a tendency just to go on and on and on and on, and it's a little tough to make your way all the way through it. But again, it's remarkable stuff, because here's what he starts with. He says, look, when I think about this origin issue, he says, it's possible, it's obvious, he thinks, that the germ of organic life came forth once at least out of the strife of the inorganic elements. But the question is, has this process occurred more than once? And this is still a debate you may have heard a number of times, right? That is since there had to be an origin of life, could there be a second origin of life? Okay? So this is where his personality comes out, and he says, if you can read this stuff, he says, since in these matters everyone has and may have his own proper fantasies, why shouldn't I have mine too? So he says, I hold the assumption that repeated creation is superfluous and not to be admitted, that it ain't gonna happen, okay? So he starts out with a point of view that for whatever complex reasons, it's too hard to do, maybe, that you couldn't have a second creation, a second emergence of life, okay? But he also is aware of the sense that there must have been some sort of history, because although this is a very strange kind of set of drawings, this is a series of fossils that he has seen. And he points out that these fossils all have something plant-like about them. And he manages to find this wonderful quote, which I assume is from Goethe, which is that all nature feels the secret power and through eternal change obeys up from the deepest region creeps the trace of life of former days. In other words, there's a history. That's what he gets out of this poem. But more significantly, he makes his own statement about this sort of stuff, okay? And what he says is here, we know that varieties once formed, and he's starting with, plants and corn and some of the things we know, that varieties once formed when they've continued to grow under the same conditions, switch in some way to subspecies, that is, into varieties which can be propagated with certainty, that is stable by their seeds. So we can have peas and cabbage, and we can get strains of those things that happen as we grow them, okay? So he says, how about if the same sorts of things that cause about these changes from the original form continue to act in the same way, not for centuries, not even tens of centuries, but for 10 or 100,000 years? Won't some of that variety become a subspecies? And so we will see them so permanent that we might actually describe it as a species. Then if we don't even have the early ones, we might consider these as unique to themselves. So that he can then say, it may be that the whole wealth of the vegetable kingdom may have been formed by gradual passage from it through varieties, subspecies, and species. This ought to have some sorts of resonance to Darwin, right? I mean, this goes right back or parallels a lot of the ideas that Darwin had, except what's missing is the selection component. But what he is showing here is the idea that there's continuous change over long periods of time, based primarily on the idea that there are fossilized forms of plants, which are not necessarily the same as the ones we have now. And so I'm not the first to have seen this stuff. There's a wonderful webpage run by a lab at Harvard, and this is the link to it. They include this fascinating, to me, fascinating extra story that apparently Darwin had written The Origin of Species. Remember when? 1859, just to get a sense of the timing. 1859, this book comes out 10 years earlier than that, okay? So Darwin was working on getting a translation into German of his Origin of Species. And this is wonderful. He had a translator, this Heinrich Braun, to whom he said, you know, I have a lot of trouble reading German. And so I don't know if anyone else has thought of these sorts of things. If they have, would you please insert this material, insert a footnote into the preface to the German edition of The Origin of Species so that we can incorporate that here. Apparently this didn't happen. So even though Schleiden's work was eventually published in English, Darwin was not aware of it, and it was never cited by Darwin as a precursor to Darwin's own ideas about evolution. It's kind of an interesting bit of history. As it turns out, after Darwin's book was published, Schleiden became a very strong advocate of Darwin's point of view. So let me then add in here a case where Schleiden was wrong, okay? And what Schleiden was wrong about was the role of the nucleus in plant development. He described it more than showed it, more than drew it. But his basic idea was this: if you have a plant cell, and he did understand that the wall of the plant was somehow extra strong. He didn't quite understand the membrane structure in a plant. What he saw was the wall and the nucleus. Okay, and sometimes, I suppose, he also was able to see a certain kind of, internal structures, but not much. Okay, what he suggested happened, and he based this on his observations in many different plants. He spent a lot of time looking at the growth of the pollen tube and the ovum, the ovule of plants, but I'm going to keep it at this simple level. So I'll draw this plant again. What he said was that the nucleus, he thought, tended to grow. So it got larger and flattened out a little bit. And then the nucleus in some way developed a little bubble. That's why he called it, remember the word he used, was cytoblast. Okay? And his idea was that this cytoblast then go back to this thing. Here's the nucleus. Here's that cytoblast, which grows and eventually splits off to be another cell. So here's the original nucleus. Not clear where the walls here are, right? But the idea is that this second structure here now expands to become itself a new cell and presumably generates a nucleus with it. And then this thing builds its own walls like that. So this is what can you do? He didn't get everything right. But as a person who established this very strong feeling that cells were the essential component of plants, that was really the critical aspect of his observation. In the botany as a public science, in the discussion of microscope technique, he says that the only good that he has seen are those made in Germany. Okay. At this point, the translator puts in an asterisk. So this is a comment, and I'll have to read it because it's too long for me to type. He says, this is the translator says, I should not, I think, be doing justice to my countrymen, that is the British, were I to allow the above remarks to pass without comment. Since the time the above was written, many improvements have taken place in the construction of the microscope, and in no country in Europe have so many of them been made as in England. Even at the time the author wrote, instruments had been made by the great English makers, Ross, Powell, Smith. You may have heard Ross's name last lecture, which have certainly never been surpassed if they've been equaled by continental makers. Dr. Schleiden's observation on English observers is, I fear, the reverse of the truth. We have many microscopes, and those the best in the world, but we've had but few observers. Our microscopes have been used rather as playthings than as instruments of profound philosophical research. Let us hope, however, that this reproach will soon be wiped away. So I think this is a wonderful little insight into what you see as the interactions that go on that we really haven't given much thought to in terms of international science. So this is a case where the translator, who's doing as faithful job as he can in conveying everything that this author had done, finally says, I've had enough. No, we're really doing a lot better than that, than you think. So that'll be it. Okay, bye-bye then.